Before we start the podcast, we wanted to let you know about WestOS. If you're a teacher or pupil at a school in Scotland, then you now have access to hundreds of quality assured video lessons created by teachers to support your remote learning. You should be able to find WestOS within the app library of Glow, where you can add the tile to your own personal launchpad. Click on the tile and you'll have immediate access to stream lessons in every area of the Scottish curriculum, with video lessons already available for biology at National 5, Higher and Advanced Higher. Now let's get on with the podcast. This podcast is sponsored by Lecky Textbooks, Revision Guides and Practice Papers, everything you need to learn, review and prepare for your senior phase assessments. Browse the books at www.leckyscotland.co.uk and get 40% off using the discount code LeckyPodcast40. We're thrilled by the overwhelming support for our episodes featuring top-notch experts delving into various biological realms. Thanks to the feedback from higher biology students, we're launching shorter, punchy revision episodes. Get ready for quick summaries of key areas to turbocharge your exam prep. These short episodes can support your exam preparation and are not intended to replace other important aspects of your revision. In this short revision episode, we are going to be looking at cellular differentiation. Imagine this. Inside each multicellular organism, including us, lies a bustling metropolis of cells, each with its own unique role and purpose. Cellular differentiation is like the master architect, sculpting these cells into specialised superheroes, each equipped with their own superpowers to keep our bodies running smoothly. Cellular differentiation is the process by which a cell expresses certain genes to produce proteins characteristic for that type of cell. So, for example, a red blood cell expresses the hemoglobin gene to produce the hemoglobin protein in order to be able to transport oxygen. The process of differentiation allows a cell to carry out specific functions. In animals, undifferentiated cells are known as stem cells, and in plants, undifferentiated cells are known as meristems. Stem cells can divide or self-renew to make more stem cells, or they can differentiate to produce specialised cells. Meristems are regions of unspecialised cells in plants that can divide or self-renew and or differentiate. Meristems are the region of a plant where undifferentiated cells can be found. And there's two types that we need to know about. Apical meristems are found at the root tip and shoot tip. They increase the length or height of the plant. And lateral meristems, also known as cambium, are found in vascular bundles between xylem and phloem. They increase the thickness of the plant. Each year as new xylem is produced by the cambium, an annual ring forms in perennial plants. You may remember counting these on tree stumps when you were younger to figure out how old a tree was. Within the meristem regions, undifferentiated cells firstly undergo cell division. Then those cells elongate before differentiating and forming specialised cells as part of permanent tissue. We learn about two types of stem cell in animals, embryonic and tissue stem cells. Tissue stem cells can sometimes be referred to as adult stem cells. Embryonic stem cells are pluripotent and can differentiate into all the cells that make up the organism. Tissue stem cells are multipotent, differentiating into a more limited range of cells only found in that particular tissue, and are involved in growth, repair and renewal of the cells found in the tissue. Stem cells can either be used for therapeutic or research purposes. Therapeutic uses of stem cells involves their use to repair damaged or diseased tissues or organs. Examples include corneal repair and the regeneration of damaged skin to treat burn victims. Many candidates struggle to explain the difference between therapeutic and research uses of stem cells, so remember to link therapy with treatment of patients. When it comes to research purposes, stem cells are used as model cells to study how diseases develop in a cell and how effective new drugs are. Stem cells are also used to study cellular processes, including cell growth, cellular differentiation, and gene regulation. The use of embryonic stem cells requires the embryo to be destroyed. It can be argued that this is the destruction of a potential human life. Stem cell use must be strictly regulated to ensure that they're not being used for unethical purposes. It is possible to expose adult stem cells to certain protein factors which induces the cells to return to an embryonic state or fully undifferentiated. The use of these induced pluripotent stem cells, or iPS cells as they might be called, may remove the need and controversy over the use of embryonic stem cells. Cellular differentiation is a marvel of nature, driving growth, development and regeneration in both plants and animals. As we unlock its mysteries, we must navigate ethical considerations and harness its potential for the betterment of medicine and scientific discovery. So hit subscribe on whatever platform you get your podcasts, so you don't miss the rest of our short summary episodes, nor our regular episodes with leading experts. And remember, this quick summary is intended as just one element of your broader revision plan. There are lots of resources available via Glow, such as Scholar or West OS, as well as BBC Bitesize. Or why not check out one of Lecky's great revision books and use the code LeckyPodcast40 to get 40% off on their website www.leckyscotland.co.uk.